السلام عليكم. أنا ريناد الموسى، طالبة الطب في السنة الرابعة. بشرح لكم lecture concepts of genetic replication and pathogenesis. Okay, first we'll start with the virus replication. A host cell acts as a factory. Why? Why is it a factory for the virus? It's literally a factor for the virus replication because it has substrate, enzymes, energy, machinery, everything that the virus needs for it to replicate and produce more and more viruses. Nucleic acid of the virus provides the genetic information. So what provides the genetic informations? Nucleic acid. It could be DNA, RNA, replication steps, adsorption, penetration, and encoding, eclipsy, synthesis of virus, nucleic acid and protein, assembly, and release. You can revise here after we finish the lecture. Adsorption is very, very and highly specific. So viral surface structures like virus-associated proteins, VAPS, or ligands bind to the host receptors. Okay, like hemagglutinin on influenza V attached to GP receptors on respiratory epithelial cells. So these virus-associated proteins and ligands are very specific. So for influenza virus, it only attack the epithelial cells of the respiratory tract, and like here for glycoprotein spikes of rabies, here the glycoproteins it only affect neuromuscular junction, and for the HIV GP120 of the HIV only affect CD4 of the immune system. Okay, I hope you understand. The main point here is very specific, and here is the examples. Here, attachment is very specific. It can be species specific. Mostly it's species specific and could be cell type specific, as I said before, like polio only affect intestines, nerve cells, but here in species specific, like smallpox, polio, measles, only affect humans. It cannot affect animals. So, the species or cell type and some are not specific at all, like West Nile virus, affect everything. Penetration and encoding. We have viral pexes and penetration. Okay? The viral pexes entry of the virus into the host cell, and the penetration, the genome enters the host cell. Okay? This is for non-enveloped viruses, and this is for enveloped viruses. Encoding, the virus nucleic acid is released from the capsid. So yes, the virus was in a capsid when it entered the cell. Here is the cell, okay? Here is the virus. It entered. When it's inside the cell, the coat outside leaves the virus. It leaves the virus. Only the nucleic acid will be left. So yes, here, this with the capsid. Now it's without the capsid, okay? This is with, and now it's without. Yeah, it should be without inside the cell. Non-enveloped viruses, endocytosis, and for enveloped viruses, it does fusion. And here, the entire virus is engulfed and enclosed in a vacuole or vesicle. But for fusion, with the plasma membrane releasing nucleocapsid into the cell cytoplasm. Here it means that the entire virus is engulfed, like it's mixing with the cell. But for enveloped, it fuses with the cell membrane. It's not entirely engulfed in the cell. Okay? So here, the cell. For non-enveloped viruses, the cell swallows the whole virus. But for enveloped viruses, the virus coat merges with the cell, and inside goes in. Yes, but the coat is like on this the plasma membrane shows part of it. Encoding may occur at cell surface. Of course, this is the uncoating. It could be in non-enveloped viruses and involved viruses. At the cell surface, for bacteriophages. At cytoplasm, for polio. At the nucleus, herpes virus. Here you can revise also after you finish. Eclipse phase. Once uncoating occurs, eclipse phase starts. What is eclipse? It's when the virus cannot be detected inside the human body. Okay, it's the period after the penetration during which no infectious virus can be detected inside the host cell. I think this is very obvious and easy for eclipse phase. Yes, so it cannot be detected at all. During this phase, the cell is directed to synthesis of viral components. Eclipse phase lasts until the first new viruses particles formed. So when there's a new virus formed, here it will be detected, okay, when it's formed. But when the viral components are being synthesized, nothing can be sensed. But when the virus is finally formed, everything gonna show in the blood test, etc. Here we have synthesis of the virus, nucleic acid and proteins, okay? So I have my strategies we will see later. Nucleic acids may be made in nucleus or cytoplasm. Nucleic acid acts as a template for production of progeny. Okay, so yes, the nucleic acid, it acts as the template. You should focus on this point. Okay, for the production of DNA, RNA, cytoplasm, RNA, nucleus, it's DNA. Okay, except for some. Okay, protein is always in the cytoplasm. A viral nucleic acid gonna be turned into mRNA, then it should be turned into the protein, which is gonna be part of the virus. Okay, early proteins, the enzymes, and late proteins structure proteins as capsids. The mRNA transcription varies depending on the type of the nucleic acid. Double-stranded DNA, single-stranded, positive, negative, double-stranded RNA, double-stranded DNA virus replication. Here we have a DNA double-stranded. We should make more DNA. So yes, copies make more DNA and leaving it inside the virus. Some of the DNA, double-stranded DNA, should be turned into an mRNA to make the viral proteins. Viral proteins for what? To cover the DNA. Yes, this is the new virus. We should make some of the double-stranded DNA into an mRNA to make viral proteins. How should we, what can we use to make an mRNA? This one, transcriptase. Okay, we should use the transcriptase, the DNA dependent one. For single-stranded DNA, for single-stranded, we should make another strand. We should make it double-stranded DNA. One strand should be used to make more copies. We should make more copies here. Okay. So this new one, this light blue color, should be used to make more of the dark blue color. So this is like the template. This is like the template which we'll be using to make more of the blue one. And now the blue one, the dark blue, will get into the virus. The double-stranded DNA, as we said here, gonna be turned into an mRNA to make the viral proteins by using DNA-dependent RNA polymerase, the transcriptase. This is how we make the single-stranded into a double-stranded to make the replication. Here, positive single-stranded RNA. For positive RNA, we should make, since it's, yes, here. So positive single-stranded RNA is it's equal to mRNA, okay? So directly we will use it to make the protein which be using to cover the virus. That positive RNA should should be turned into negative RNA. Why? For it to be used to make more copies. More copies. To make what? The positive RNA. Because we use it as a template. Now we will have more of the green RNA. Negative single-stranded RNA. Here we should change the negative into a positive. It's not gonna be a positive RNA only, it's gonna be positive mRNA. Okay, RNA dependent RNA polymerase. This is what should be using to make mRNA. So here, as we said, it's gonna be turned in this pathway to a template. Here it's a template which we will be using to make more copies now inside the proteins. Yes, need to be a positive RNA, so the negative need to be turned into a positive RNA. Here for double-stranded RNA virus, it cannot be translated directly. It need first to be positive RNA, then becoming the original double-stranded RNA. And this is gonna be the viral protein, of course. For the retrovirus, here the positive RNA gonna be turned into a single-stranded DNA, then becoming double-stranded DNA, then becoming RNA. This is weird, I know, but it's retroviruses. So yes, it gonna be turned from a positive to a single. It's from RNA to a DNA, becoming double DNA, then turning like the first step, positive RNA. The double-stranded, of course, gonna be turned into mRNA to make the viral protein. Here also retro, so yes, double-stranded, so it's gonna be acting in this way again, becoming positive RNA before becoming the single double. Yes, it's doing it the same, not like the previous viruses. Like here, double-stranded directly inside the virus. Here, no, it's gonna be going into a long pathway. RNA, single, DNA, and the double, as I said before, mRNA, protein, covering the virus. Assembly and maturation. Assembly, it's also called morphogenesis. Assembly of nucleic acid and protein coats to form a mature virus occurs in nucleus. Nucleus for herpes viruses, and cytoplasm for polioviruses. At membrane enveloped viruses. I have a simple way to memorize the three. I don't know if it's gonna be helpful for you. At membrane, imagine a house and the mailbox outside the house. So at the membrane of the house, there is gonna be an envelope box for the mail, envelope for etc. In the cytoplasm for polioviruses. So yes, in the O, imagine the cytoplasm being all round. In the nucleus, it's herpes. No O, so it's inside. It's like a small dot in here, so it's gonna be nucleus. This is my way of memorizing these three. Structure of protein, capsmeres, which aggregates, forming a procapsid. So yes, what is a procapsid? It's just capsmeres, all condensed. So yes, capsmeres equals a lot of capsmeres equals procapsids. Capsids, of course, no nucleic acid, it's pro. So a nucleic acid inserted through a pore in procapsid, then a structural reorientation of procapsid leading to internalization of nucleic acid. So there is a pore in the procapsid. So yes, like here, there is a pore it's now getting inside. Then there is structure reorientation. So it changes the shape of the covering to make the nucleic acid trapped. Structure reorientation of the procapsid leading to internalization of nucleic acid. The capsomeres accumulate accumulate. So it didn't just change shape. Now it's all condensed, very condensed. Capsomere accumulation around condensed nucleic acid was being synthesized to form nucleocapsid. So, after assembly, some viruses stop here at the assembly, some are not, like enveloped viruses in these envelope. Non-enveloped viruses fully developed virion, but for enveloped viruses, it has only the inner part, the nucleocapsid. Here is just the simple thing for these 3 points, okay? You can read it after you finish these main 3 to make it easier. Now, in maturation for enveloped viruses. So the envelope, it can be from the host cell membrane. Yes, it can be from the nucleus or the cytoplasm. So envelope from the host, not from the virus. For the one assembled in the nucleus, it comes from the nuclear membrane, but it transported to cytoplasmic membrane in a vesicle and then fuse with the with the cytoplasmic membrane. So here, the virus assembled in the nucleus, like herpes virus, is having his membrane from the nucleus. Then it transports to the cytoplasm and fuse with it. For the virus assembled in the cytoplasm, like orthomyxo and retrovirus, it comes from the cytoplasmic membrane during budding. So yes, it starts in the cytoplasm, stays in the cytoplasm, but for the nuclear, it comes from the nucleus going to the cytoplasm. Enveloped are modified by adding of glycoprotein. When we have glycoproteins, it can infect directly. So, here, the release. Lysis of the cell, cytolysis, mainly with non-enveloped viruses, released at once. But budding through plasma membrane exocytosis, mainly with enveloped viruses. So, lysis, non-enveloped. Budding is for the enveloped viruses, where they require lipoproteins enveloped during budding from the outer membrane, released slowly over time. This is how it's released. Lysis for non-enveloped, it's released at once. Budding for enveloped, released slowly over time. Here, not every release variant is infectious. Like here, the envelope viruses, if it has glycoproteins, it will be infectious, but if it doesn't, it's not infectious. Yes, so the variant can be infectious or not. In varicella, they infect by direct transmission between cells. This is how they infect in varicella. Properties of enveloped viruses. Generally, they are environmentally liable. Envelopes is sensitive to drying, heat, detergent, acid, solvent, as ether. Must stay wet during transmission. So, if the virus is on a table, it's not going to be transmitted. It should be wet, transmitted in large droplets and secretions. It cannot survive in GIT, okay? Because of acid, it's acid liable. It doesn't need to kill cells in order to spread, okay? It doesn't. need to kill the cell in order to spread. May require both a humoral and cell-mediated. Properties of naked viruses, the non-enveloped, okay? Non-enveloped. Generally, they are environmentally stable, so they are stronger even though they don't have an envelope. So, capsid is resistant, drying, heat, detergent, acid, proteases is resistant to everything. Retained infectivity on drying. Survive well on environmental surfaces. Can survive in GIT. So, surfaces like a table, like skin, it's okay. It's gonna be it can be spread easily via vomits, hand to hand, dust, small droplets, everything. Must kill host cell in order spread. Require only HMA to neutralize infection. Now we'll start with bacteriophage. Bacteriophage is a virus. Yes, it's a virus that infects bacteria. It's well distributed in nature. Has a role in transduction. Has a role in phage conversion, cloning, controlling bacterial colonization, phagotyping. So, for transduction, move gene between bacteria. So yes, the virus goes into a bacteria, taking the genetic information, going to another bacteria, and putting it in, okay? In phage conversion, it changes the bacterial trait, okay? So yeah, it gets in, changes the bacteria, then leaves. Cloning vector, used in research, in gene research. So yes, because it's have the cloning feature, can be used in gene research. Control bacteria population, decrease bacteria in water, phagotyping, identify bacterial strain by the phagotyping. Morphology, it's formed of head and tail. The head has a double-stranded DNA. This is important. Have a double-stranded DNA core and protein capsid. Double-stranded DNA core, protein capsid. The tail is a hollow core, contractile sheath, and a basal plate. Here, the head having nucleic acid, which is a double-stranded DNA. Double-stranded DNA. Here, we have the contractile sheath, and here is the base plate and the tail. Here is the tail, obviously. These are the important points. Some phages are spherical, filamentous, and it can also have single-stranded DNA, RNA. For the life cycle, it can be of two types, lytic or lysogenic. Lytic is multiplication, leading to lysis of the host cell and release of the virions. So yes, it lyses the host cell and then it's released. For the lysogenic cycle, it integrates first into the host genome. Okay, for this, I have put in here a video. You should watch it. It's very helpful. And here, you can see how the lytic and lysogenic cycle are mixed. Okay, it's very easy. Just watch the video. And here just you should literally just memorize it. The key points, the important points. Yes. For any question, please contact me on this email.